Okay, Biology B students, here we go. Tools of classification, finishing up our evolutionary module. Some of the things that you should be able to understand by the time you're done with this particular presentation / lesson is understanding and interpreting evidence such as cladograms, dichotomous keys, biochemical relationships, for example, the DNA amino acids, morphological, structural similarities to identify organisms, and lineage relationships, or following paths of similar organisms, or when they branch off and become a little different from each other. Compare and contrast the characteristics used to classify organisms into the domains, and then whether or not they're plants, animals, fungi, bacteria, so forth. Okay, so modern taxonomy has a couple different things that allow a taxonomist, somebody who is going to study classification, to look at and identify and differentiate between creatures. So, for example, you have homologous structures, right, where structures are going to be similar amongst certain groups of creatures. Embryonic development, or embryos, looking at the embryos and seeing how similar or different they are. Molecular similarity of amino acid sequence of proteins. So if you look at a particular protein that we have and that, say, a rabbit has, how similar are the sequence of amino acids? We'll look at a cladogram, which is just a tool and a chart, and then a dichotomous key and how to figure out things. Use dichotomous keys every day of your life when you're making decisions and trying to categorize things. So, have you ever wondered who your ancestors, who they are? How far back can you trace your family heritage? Lineage, descent from an ancestor. In this module, we will look at the lineage of organisms and the tools of taxonomists use to classify them into different groups. Same structures. So if you look at the picture on the right, it's the best way to look at this particular term, right? This homologous structures. If you look at the human, if you look at the horse, the bat, and the porpoise, they all have three main, actually even four, main areas that are similar, okay? If you look at the humerus, right, even though the size and shape might be a little bit different, it's still in the same basic location. The idea is to connect the lower appendage to the shoulder area, all right? That's your humerus. Then underneath that is the radius ulna, right, where you have the humerus that is connecting the fingers, all right, to the humerus, and that's the radius and the ulna. And if you look at the little yellow bones, those are the bones of the wrist. And then finally, you have your finger bones, right, your carpals and metacarpals and all that stuff. So they're all similar, all right? So similar structures. Homo means the same. So homologous bone structure allows a classification specialist to look at and go, they must be of the same lineage as far as arm bones go. So same structure, different function. Notice how the bone structure is very similar, but yet the limbs all have very different functions. All right, so our fingers versus the metacarpals and carpals of the human versus the bat or the horse, very different, and even the porpoise. So, but then anyway, they show a little bit of common ancestry. We look at another tool, embryological development. If you look at the picture again, from a very first few stages, many of those embryos look very much the same. And then as they start to differentiate, they start to change. And now you can see a little more like what they would look like as an adult to some extent. But prior to the developing embryo, the very last stage, it is difficult, all right? And they have many of the same structures. So early development is similar. Number of gill arches, almost the same. As the embryo develops, chemical signals change the embryo to a specific species. You can watch the video and learn a little bit more about embryological development. Another tool is molecular similarity in the DNA, RNA, amino acid sequence. What do I mean by that? Look at the picture. All right, a particular sequence for a particular gene, from a particular gene for a particular protein has the DNA sequence for a human and the chimp, and you look at those sequences, they are identical. And so same sequences of G's, C's, T's, and A's, all right? And therefore that protein that will be produced is going to be the same. So the analysis of our DNA and other species show a remarkable amount of similarity, and it allows scientists to use DNA sequences of other creatures to help humans deal with some different diseases. So we have the ability for humans to use insulin that's created by pigs to help with diabetes. A cladogram is another tool, right? Here's a cladogram. You look at the picture there. As you go up to the right, you are showing differentiation. And as you go higher up, then you're looking at time. So the y-axis, kind of think of it as a time thing. The x-axis, if you wanted to, or to the right, is a change or a little difference between the creatures. So if you look at like the hagfish, it's very different than the chimp. So in the cladogram, it's a diagram that depicts evolutionary relationships among groups. It is based on phylogeny, which is the study of evolutionary relationships, sometimes called a phylogenetic tree, although not accurate. So if you look at this picture here, the kangaroo and the koala are very much the same, are very similar, okay? Way more similar than the bat and the lion. And so as you go to the right, all right, things will be being passed on, while if you just stay in one area and you stop as time goes on and differentiation occurs, the kangaroo and the koala become different than the bat and the lion. So you can read through this, but basically they're all mammals. But as time went on, right where there was a split here, the koala and the kangaroo have similar features, but yet the bat and the lion continue to evolve and develop and have different traits than the kangaroo and the koala. All right, so we talked about your closet not too long ago. Let's just say you just threw all of your clothes on the floor, okay? And you started to classify them and put them into your closet. Well, what would some of the things you would ask yourself? Okay, so the first question would be, is it a shirt? The answer is going to be yes or no. If it's a shirt, okay, we can put it in a pile. And then you can further ask yourself, is it a long sleeve shirt or a short sleeve shirt? You can split those up. Then you can ask another question. Does it have stripes or is it a solid color? Okay, you can split each of your shirts up into solids and colors, and your short sleeve shirts, long sleeve shirts into stripes or no stripes, and the short sleeve shirts into stripes and no shirt, long sleeve and no short sleeves. So as you continue going through it, you're basically asking these questions, and then sooner or later you have your whole wardrobe classified. If you look at the dichotomous key question, or the picture on the right here, has pointed ears or rounded ears, and then you can go on to the next question. Has a tail, no tail, ears pointed upward or downward. So dichotomous means it's basically a set of two questions. It is or it isn't. It's this thing or that thing. The questions are designed to find or eliminate characteristics of an organism. Broad questions become more and more specific. So your final question could be, it can be really specific into colored or white and black, something like that. So here's an example, all right? Tentacles. If it has tentacles, go to question number 2. Does it have 8? Or does it have more than 8? All right, so then you look at the bottom right-hand picture though, the orange anemone. Right, it has lots of tentacles. So if it has more than 8, then it says go more than 8 tentacles, go to question 3. Did it hang down or the tentacles are upright? Okay, well, if they're upright, then it's anemone. If it's hanging down, you got to go to 4. Balloon-shaped body, could be a jellyfish. or it's not. So there's your tools of classification, lesson 3.3, biology B. Good luck.